So you guys just had exam 1, and I think I posted exam 1 results. So there was a high of 94, the mean was 64, and the median was 68. The median means half the class got 68 or above, half the class got 68 or below. And that's not bad because 70 is about the C range, right? And so what I mentioned to you guys is that the class is graded on a curve. So basically, if you score 90 and above, you know you've got an A, right? And 90 could be an A minus. It might be a solid A depending on the curve. If you score between 80 and 89, you'll get nothing less than a B. But again, depending on where the plus and minus ends up falling, because it depends on where the curve was. So a traditional 80 would be a B minus, but with a curve, an 80 could end up being a regular solid B. So the people that benefit the most from a curve are those in the C range. So for example, a C for this class based on this one exam, if we just did it off the one exam, would be the mean, and the mean was 64. So 64 means that was average, that was right on the C, that was the top of that bell curve, okay? So as long as you scored near the mean, you are in the C range. If you're trying to figure out, like, where did you land with the rest of your peers in here? And so I encourage you to try to be closer to a high C range because, or higher, you know, obviously. But a C plus would mean that when you're trying to get into one of the other programs, you would be a better candidate than the person that has a C, for example, right? So a C in this class at NIC is like getting an A somewhere else. So, you know, I'm not going to name schools, but basically, you know, you get an A in one of those classes, they know a C here is like getting an A over there. So some places grades are inflated, you know. Your grade is not inflated here. You work very hard for your A or your B or your C in here, okay? And so, and when they are accepting you or looking at you as a candidate for their programs, they know how we test you. They know the rigors that you've been put through in this course, both in lab as well as the lecture exams. And I will tell you, student after student that has gotten into, for example, the nursing program has come back and told me that the way that we tested and prepped them in AMP 227 and 228 really got them prepared, and they didn't struggle as hard as people that came from other places that got into their program. And they felt that they were well prepared for what we're doing. You guys sometimes feel like we're mean or, you know, or, you know, seriously, why are you asking it that way? Or why do you have to tie me and stare at me in lab? But any of those programs, that's what they're going to do. You're going to have time tests. You're going to be, bless you, you're going to be looked at where you got to do it within a certain amount of time. You got to name it. You got to be specific. If you're planning on going into the healthcare field, you need to be specific. You can't be general. Okay? General is what gets mistakes. And you don't want the person making mistakes working on you, I'm sure, right? So you want to be good through your program so that you're even that much better when you're done with your program. You want to be the kind of person you would want working on you. So most of your exam packets were given back on Thursday evening or Friday, but internet students can print out their exams in their Blackboard, or you can get a blank copy from me up in the across from the lab in my office. And just like in the blog post, I mentioned a couple things that you guys can do if you didn't get the score you were hoping or intended to get, or you got a good score, you just want to get a better score. There's a couple different things that could have happened during the exam. One, maybe you just had a little test anxiety, right? So we suggest that you take the exam home without trying to look up all of the answers and figure out what would have been a right answer, retake it. Use like a different colored pencil or crayon or whatever, I don't care what you use. But go back through it again, take it again, and see if you would have gotten a better score. If you did, there's a couple different reasons. Either one, there wasn't the test constraint, you weren't freaking out, and if it's an anxiety issue, you got to figure out what's going to work for you to calm you down, not have those nerves, that kind of thing. Otherwise, it could have been that, you know, if you found that you marked a bunch incorrectly, and when you went back through it and you got it right, maybe you didn't read the question carefully or all the way through. You got to read every answer. So there's, you know, 3 out of the 5 answers are usually pretty easy to knock out of the way and say, nope, nope, nope. Haha, you tried to trick me. Yeah, right. You know, 2 of them might be a little difficult to discern, and on some of them they're really obvious. There's only one right answer, but sometimes there's 2 that look like, ooh, I'm not sure which one it could be. And so that could be a content knowledge thing where you thought you knew the information, you just didn't know it to the detail that you needed to know it. You didn't realize that you had to know every single teeny weeny little detail, right? And this is very much a detail-oriented class, both here and in lab. The other thing could be that you just read too quickly and you knew the answer and you just weren't careful when you went to mark it, right? So if you know that, then next time you take the test, you know you need to slow down. Check yourself. If you went through and you second guessed yourself and ended up picking the wrong answer, so you went back and you changed your answer, stop doing it. Go with your gut. Okay, so those are some things that could help. Now, if you did okay and you just want to do better, you know, keep doing what you're doing and maybe add to whatever it was that you tried for your study skills. If what you did didn't work at all, you need to change it now. Don't wait till after the second exam. Don't wait till the 4th exam. Change it now. If you got a grade that you feel was horrendous and you're thinking, oh my gosh, what happened? Don't do it again. Don't do the same way you're studying. I mean, you may get the same score, but change what you're doing. Don't wait. Okay? Get into small groups and study. Do a tutoring thing. If you were in a small group and it didn't work, maybe the dynamic wasn't right. Too many people there. Maybe the person that you were trying to study with, it just wasn't a good mesh. They're a nice person, but it's just not right for you. It's okay. Don't be offended, right? If you didn't verbalize it at all, you should say it out loud. Sherry highly recommends rewriting everything. And I posted then what another 228 student said that they did to change what they were doing. I had another student that got somewhere in the 60s on his first exam. And so he immediately changed what he did and he got 90s on all the exams after that. It can happen. But you got to figure out what worked and what doesn't work. And don't keep doing what didn't work and expect it to work better next time. Okay? If you're not sure what else to try, study habit-wise, come up and see me. You can post on these blogs and say, hey, I need some help with some study skills. Anybody got some suggestions? You know what I mean? So make the changes now, because remember, exam 6 is 10 questions from each of the first 5 exams, right? So start, after you figure out how you did and where your pitfalls were on this exam, then go through and start memorizing exam 1. There will be 10 questions, identical. They might be jumbled up within each other, like the answer is A is now a D answer or something like that. But it'll be worded exactly the same, no changes. 10 of them, I'll randomly pick them from exam 1, 10 from all the other exams. And then if you start memorizing them now, you can get 100% on exam 6. So if this was your poor score, it will replace that score, which is wonderful, right? If you did okay on this one or you did poor on this one, don't expect, something could happen. Life situations happen. You get sick. People have been getting flu like crazy, right? So you don't want to anticipate that, oh, it'll be fine. Exam 6 is going to take care of this. I don't have to worry so much about my other exams. Study like your life depended on it and on every single exam, because you never know when you're going to get sick or something happens, okay? Any questions on the exam, study skills, anything like that real quick? Okay. Another thing that you guys may not have realized, but if you go up into resources, you can click on the curve, and there is a short video there that Peter Zayo narrates and explains to you how the curve works. So you can kind of see how they shift the curve. And the curve isn't set. It's not like each individual exam gets curved. It's after exam 5 is when the curve gets set. But if you use the mean and the median to kind of see where you were sitting in the class, you'll have a pretty good idea. I had a student before, they were consistently getting, I can't remember if it was 40s or 60s, like low 60s on every single exam, and they didn't understand why they didn't have a B at the end, like a solid B, and I'm going, how do you think that score, and if people are getting A's, it's not going to get curved that much, right? And people do get A's. People were getting 94 and stuff, right? So you can't expect what would typically be a D to all of a sudden magically appear as a B at the end of the semester. The curve isn't going to curve that much. It's going to help a little bit. But I want you guys to understand, because it's at the end of the semester that you guys start freaking out about where you are and how does the curve work. Now you know where to go for the curve. You can watch that video, And I always post, the high, the mean, and the median after the exam scores are all in, okay? Another thing is you can scroll to the bottom of when you're on the home page, and at the very bottom, you'll see there's the old exams, but there's also current exam keys. So you can click on that. It brings up the exam. If you're an internet student, you'll have to look in your Blackboard account if you want to see exactly which questions you got right or wrong. But if you get a blank copy from me, you just look, just like the paper exam students, where you just look at what version you have, and then you click on that version, and that's the answer key. Okay? All right. Where did we leave off? Let's see. We talked about this translation. So we did a little bit of transcription, right? All right. So let's see. Oh, the other thing I was going to say, please don't listen to the lectures when you're driving. You're just teaching yourself to fall asleep in one ear, out the other. You're not going to get it. If you are going to re-listen to any of the lectures before an exam, you need to actively be participating in that listening. Like have your notes out in front of you, adding to it, things that you might have missed the first time around. So I just wanted to put that out there because I had somebody tell me one time that they were listening to me all the time, and I'm like, no, that's not helping you. Don't do that. All right, so let's see. Back to our notes. We're going to finish transcription and translation and then go on to cells, and then chapter 5 is tissues. And so did you guys realize that we're following the structure or pattern or format that we talked about from chapter 1? So we went from atoms and molecules to cells and then tissues, and then after that we go into organs. So we're kind of following the same pattern. You guys are learning and building on. That's why some of what we talked about, bless you, on last Tuesday was a pretty good review, of exam 1 material. And you'll see that we keep talking about some of this stuff as we go. So we're talking about transcription. It occurs where? Transcription, the first part, occurs in the nucleus. And we make mRNA from DNA. We modify the mRNA so it won't get eaten up by nucleases, which are in the cytoplasm, right? And then we send it to the cytoplasm, that messenger RNA, which is where we're going to do translation. So remember, we're like translating another language. We're going from a nucleotide language to an amino acid language. And so this occurs at the ribosomes. And what are ribosomes made out of? rRNA. We have a large ribosomal subunit and a small ribosomal subunit. They will come together, and the messenger RNA will be read through it, kind of like a ticker tape, like okay? And from there, we'll bring in, we barely mentioned tRNA or transfer RNA is going to bring the amino acids there. They transfer the amino acid from that tRNA onto the growing polypeptide chain. Let's see. So, which polypeptide chain is your protein, right? So we're going to continue where we left off, looking at the details now of translation. So we said translation occurs in the cytoplasm. Initiation, coming together of the messenger RNA, the ribosomal subunits, large and small, that's the rRNA, and the tRNA. All 4 of those have to come together to get it going, okay? So you can't build your protein if tRNA isn't coming in there. Bless you. So I mentioned to you guys, I wrote out a piece of nucleotide language here. Is it DNA or RNA? DNA. Why is it DNA? Because it has T in it, right? Thymine. So we are going to make, if we're translating, so before we could translate it, we took our DNA, we have to make messenger RNA from it. So let's write out what the messenger RNA message is, right? So it's complementary to this. So an A would be a U, A, U, G, C, U, A, U. Hey, you! No. G and A. Okay? That one's kind of looking funky. All right, so, and put that last one back there. Okay, so we wrote it out, and now, remember in transcription, right after we've just transcribed it, right? It's like the transcriptionist writing down the same message, same language as well. And we would have put what would we do to mature it? We would have taken out things that we didn't need. We put a cap on it and a tail on it, right? And then it would be mature. It could leave the nucleus, go out into the cytoplasm. So as we're continuing on with our story here, one of the things that you'll notice that we kind of mentioned that there's going to be something called a codon and an anticodon. So codons occur on the messenger RNA. So the sentence, it reflects a DNA complement. This is your DNA complement, the first strand that we had, right? So if you really want to call them triplets, so 3 nucleotides together would be a triplet would be on the DNA. If you have mRNA, your triplets are now called codons. So you need 3 that are covalently bound to each other, 3 in a row are a codon. So this was our DNA strand, this is our mRNA, and it has codons on mRNA, okay? So 3 sequential nucleotides on mRNA. Now, what am I going to tell you? Go through my notes. What it says in initiation where it's saying coming together of the messenger RNA, the small ribosomal subunit of RNA, the tRNA, and then the large ribosomal subunit of rRNA. It kind of comes in that order. So it's not like the ribosome was already put together. It's like the pieces are near each other, and the small piece is on the bottom, so it kind of looks like this. Then you have your messenger RNA is coming. It's going to go through that direction. You have tRNA, which kind of looks like a T or clover leaf if you want. So he kind of looks like this. His anticodon will be at this end on the bottom, and at the top will be his amino acid that he's bringing over. Then you'll have this large ribosomal subunit that's going to come and sit here, and the mRNA is going to be read through here. tRNA is going to come. There's actually 3 sites in here, so the tRNA comes to the first site, then it will shift over to the second site, then it will shift over to the last site, and then once the whole messenger RNA is read, and you end up finding out what's called where it starts and where it ends, then that ribosomal subunit, the tRNA gets kicked off, the ribosomal large and small subunit come apart. And another ribosomal subunits, small and large, can come together and hook onto an mRNA and start reading it again. And it'll keep doing this until you no longer need the protein made, or nucleases start chewing things up, or so on and so forth, right? So that's the idea of what is happening there. Now, it says it occurs at a start codon. A start codon is telling you when the message starts. Okay, so our start codons always start with the nucleotide codon of AUG. So you can think August, school starts in August if you want. That works, right? And AUG is the codon which is on the mRNA molecule. It codes for a specific amino acid. And the amino acid, this is the only one that you have to memorize, that AUG is a start codon. The amino acid that it is associated with is methionine. It's the only amino acid I need you to memorize, okay? So all proteins will start with methionine then. However, you know, when they end up going through the Golgi and stuff, and remember I said we'll modify our protein, we'll take other things off, we'll add carbohydrates on, we might, you know, change the shape, go to secondary, tertiary, quaternary structures, that kind of thing. So at that point, going through the Golgi, we'll remove those methionines later. But all of them start with methionine until we modify it. Okay, so let's see. So the codon reflects the DNA complement. So if the codon was AUG, what would the DNA complement be if we went backwards? We don't go backwards, but if we were to go backwards, what would the DNA segment be for AUG? TAC. So on DNA, it would be TAC. So if I were looking at this DNA molecule, I'd be looking for the TAC, but that's not really how we read it. I just wanted to put it out there in case you get like some sort of question where you're trying to go backwards or forwards, and it's kind of like multiplication and division. You can go forwards or backwards on a math problem and still get to the same point, right? Anyway, okay, so on codons. So if I were to look at my mRNA codon that we wrote up there, or not the codon, but the mRNA messenger RNA that we have, where would I start reading it? Where does the message start? Does it start here? 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 Yes. Okay, so my first codon is here, AUG. So I'm going to put first, bless you, codon. Okay. So since you know that that's where it starts, now you can mark off every codon. So every 3, you're going to mark off a spot. So here's the next 3. That'll be the second codon. Here's the next 3. That'll be the third. Here's a fourth codon. Here's a fifth codon. And proteins are really, really ginormously long. Lots and lots of amino acids linked together. But in this case, let's just say we have about 5 codons, right? Now, AUG is our start codon. It's on the mRNA. It's basically saying, hey, start making the protein starting here. This is the beginning, okay? We also have something called a stop codon. And so let's see if we have that on here. There we go, where it says termination. It says a stop codon is reached and newly formed protein is released. So when you see the codon, and you just have to memorize these 2, there's actually one more, but you just have to memorize these 2, UAA or UGA, so you could do UA or UGA, whatever you want to do to remember this. Those are stop codons. Stop codons do not code for an amino acid. What they really code for is something called like a release factor, which tells the growing polypeptide chain, you don't add, when you see this stop codon, you don't add an amino acid on. This is saying this is the end of the message, the end of the protein, go ahead and release. And so this would tell the ribosome to come apart, and you don't read any more on, because there could be more message on that mRNA, but when you hit a stop codon, you're done. You don't keep reading anymore after that. Okay, it's not important. It's like gibberish after that. You don't care. Okay, so can you guys see that if something happened, if you had something that happened to like one nucleotide of your amino acid code, that you could have an issue with a protein being made too much that isn't a correct protein if it didn't get the right stop codon in the right place? Or what if something happened and you get a stop codon early, right? You don't make the full protein. If the protein isn't made the way it was supposed to, it is not going to function correctly. Okay, thus like sickle cell anemia. It's one amino acid is wrong. That's it, one. And your blood cell can't carry oxygen very well. So I just want you guys to kind of notice that. So if we go back up, we have elongation is where the ribosome sequentially reads the codon. Sequentially just means it's reading it in order, because remember, it's important how we link our amino acids. That primary sequence is important, right? And then it says tRNAs are active in the cytoplasm. tRNA is transfer RNA. So that contains the opposite of a codon called an anticodon. So that's easy to remember. So tRNAs have the anticodon, mRNAs have the codon. And so that's why it says it's the complement of the codon. And these bring in, Sherry likes to use capital A's for amino acid. I use little a's. So AA is amino acid to add to the growing polypeptide chain. So what would be the anticodon for AUG of the mRNA molecule? UAC. So if we had UAC, can you guys see that? And I'm going to draw kind of a funky shaped tRNA. So let's say this is the top of my T, and it had like, 2 little arms here. And then it has like this length here, and then it's going to have an amino acid attached there. What would be our amino acid in this case? The only one I asked you to memorize is methionine. So I'll just put MET, MET is short. It's how we write some of the amino acids. So we have this ribosome. It's reading. It sees the codon for start, which is AUG, which means the tRNA that I said before is everything's always so specific. So only this anticodon will have methionine on it. And it would, temporarily hydrogen bond to the area while it's doing its thing. And then the next anticodon or the next tRNA would come into place. So who would be our anticodon for the second codon? That sucks. Okay. So we have C, G, G. Who would be our third anticodon? A, no T's though, because that would be DNA. So it's got to be a U, right? Yeah? Okay, and then what would be our next one? A-U. And what would be our fifth one? A-C-U. Okay, so I'm just going to draw it instead of the T that looks like the clover leaf. I'm just going to draw like a bubble just to be quick about it. Okay, so there'll be an amino acid there. There'll be an amino acid here, an amino acid here, an amino acid there, right? Or would there? Do you guys see any stop codons on there? Who's the stop codon? A last one. So would there be an amino acid? No. So, okay, it would end up being a release factor. So in this case, if you had 1, 2, 3, 4, 5 codons, how many amino acids long would this protein be? Only 4. Okay? Because remember, stop codons do not put on an amino acid. If your protein was 100 amino acids long, how many codons did it have in the mRNA? No? Backwards. If the amino acid has 100, it would have 99 codons. Right? Did I say that wrong?
Yes.
The amino acid has
100.
100.
You have to have 101.
101, you're right. I was making sure you paid attention. All right, so you guys get it though. So you always have one more codon because of the stop codon that doesn't code for an amino acid than you would on your amino acid chain. Does that make sense now that I said it correctly? All right, so let's see, what else do I want to say? So when these amino acids are linking up, what is connecting the amino acids? What did we create here? We created a peptide bond, which is a covalent bond, but more specifically because it's a protein, it's a peptide bond. So we are creating covalent bonds between those amino acids. And did you guys notice our proteins, they don't have tRNA hanging off all over the place, right? So we have to have a way of cutting off the amino acid from the tRNA. So we're going to cleave this off and cleave that off as we are transferring the amino acid onto the growing chain so that you would end up having your MET, whatever generic amino acid, okay, like that. You don't have tRNAs hanging off of it. So the ribosome is attached to the messenger RNA. You have the large and small subunit. That's what initiation was that we said was number 1. It moves in the direction, so it's moving along this way as it's, in this case, reading the next codon. And as the ribosome moves down, it reads the codon and it brings the complementary anticodon or anticodon, the tRNA, and that has the amino acid to that tRNA. So each tRNA is specific for one amino acid. When it lets go of the amino acid, that tRNA will get ejected and will be able to go out. And what amino acid is this first one going to go find again? Another MET, another methionine. It only codes, that tRNA only codes for methionine. The second one only codes for whatever his amino acid was. So he lets go and he goes back out into the cytoplasm and he finds another one of his amino acids that he codes for and he brings it over to another ribosomal subunit and mRNA message and keep putting on. That's all they do. Bring another one, put it on. Bring another one, put it on. Eject, put another one, put it on. That's all they do. And that is called elongation, which is number 3 here. And so ribosomes sequentially add them on. And so this adds to your elongating your polypeptide chain, and it keeps adding it until the next amino acid, until it reaches the stop codon on the mRNA, which will cause what? Stop codon causes termination, the release of the ribosome. Okay, and let's see what else do I want to tell you. That looks good. All right, so when we look at what our ribosome looks like. So on this picture, the ribosome is this yellow globby thing here. And you could remember the sites are EPA or like Environmental Protection Agency, but that's actually the backwards way. So actually the the messenger RNA gets read through it in this direction. The A site, A for the beginning of the alphabet, or APE, if you want to think of it that way, is the first site. So A is kind of like the holding pattern at first. So your tRNA is going to come to the A site that is complementary to the codon on the messenger RNA. Then the tRNA will transfer over to the P site, and this is kind of weird, it sounds backwards. It takes its amino acid that it had on it, and the next tRNA is coming into the A site then. So it's always like one's coming right after the other, okay? He will transfer the tRNA that's in this spot, will transfer his amino acid to the one in the A site. Okay, so I'm going to put a circle around this amino acid. It's going to get linked to his amino acid. And then this shifts over, the one that is now in the A site shifts over to the P site. The one that was in the P site shifts over to the E site. And you can think E for exit, E for, I don't know, I had a bunch of words. E for exit, eject, end, whatever you want to think, right? So that tRNA is the empty tRNA for E also, and that'll get kicked out then so he can go find another amino acid and start over again. Does that kind of make sense? I know this is so fun and exciting. You guys look super uber excited today. All right, so let's see. We keep doing this until we get to a what? A stop codon, which were what? UA or UGA, right? Okay, and then we don't put a codon, we don't put an amino acid down. It ends up having a release factor, and we let go. That's the story of how you make your protein, essentially. So now we can look at cell growth and reproduction. All right. And so the cell cycle or cell life cycle, we have two main phases, interphase and the mitotic phase. And so interphase ends up being the longest phase, which means your cell is typically in interphase for the majority of its life cycle. And it's only in the mitotic phase for a very short period of time. Some cells will be in a mitotic phase a lot more often. Just depends on the kind of cell and its function and what it needs to be doing. But if, let's say, it took 24 hours for us to make another cell, it would be an interphase like 23 out of the 24 hours, and the mitotic phase might only be 1 hour. So there are other cells that do it even faster than that, and ones that do it much slower than that. But in our cell, how many chromosomes do we have? 46, right? We get half of them are from mom, half are from dad. So 23 chromosomes, 1 through 23, because we just number them by their length, right? So 1 through 23 from mom and a 1 through 23 from dad. And we must replicate, or you could think duplicate, exactly that DNA, or else what happens? You get mistakes, you get problems. We don't want problems. Our DNA, we try to uber protect it, right? That's why we stick it in the nucleus. We don't let anything get to it. We don't let it out of the nucleus and that kind of thing. So we need it to be exact, identical, no mistakes, no problems. And you guys actually, we don't have to worry about it in this class, but you guys actually have enzymes that go back and reread your DNA, and it tries to make sure there's no mistakes, and it can go back and correct it here and there. So, but we're not going to get into that detail in this class. So, interphase is basically the whole blue part of our circle there. So, like I said, the majority of your cell is in interphase. This is where the cell is doing what it's programmed to do. So, if it's a liver cell, it's going to detoxify stuff. Like if you drink alcohol or you're taking medications or drugs, it's going to help filter that out and make it not so toxic for you. Liver cells do other things too, but that's just, people know detoxification. So the whole point of going through reproduction, and this isn't reproduction like having kids. This is reproduction of let's make an identical cell. Okay, this has nothing to do with children. You're not making progeny. No sons or daughters, no aliens, nothing like that, okay? Just another identical cell. If you're a skin cell, you want another identical skin cell. If you're a liver cell, you want another identical liver cell, okay? So with cell division, you end up starting with one cell, and sometimes they'll call it the parent cell or the original cell, and you're going to duplicate or replicate the DNA, and you're going to have to grow your cell so that when it splits into two, you'll have mitochondria, you'll have ribosomes, cytoplasm, all that good stuff that the cell's going to need. So you go from one cell and you're going to end up with two identical cells at the end. That's what happens. This is what mitosis mitosis is. Okay, so, and you can think of it as like they're identical twins essentially, all right? So if we look at interphase, which is the whole blue section, there's 3 parts to it. There's G1, S, and G2. This is the time from the cell's formation. So you start with your cell, maybe it was just divided, and now it's going to grow and do its thing before it can divide also. So the cell's doing what it's programmed to do. It's growing, it's hanging out, it's functioning like normal, okay? So let's see. So G1 is for growth. So this occurs right after a daughter cell is born. So this is the identical cell that has just been made. And if you're a baby, what are you trying to do? You have to grow. You need, hopefully you're a chunky little baby too, so you have reserves also, right? So it's growth. So it's smaller than the original cell. It's going to end up being the same size as the original or parent cell. S, which is in the middle here, this is where the DNA synthesis is going to occur. So we're in interphase. DNA is in the form of chromatin. Do you guys remember what chromatin looked like? That's when the DNA is not as tightly coiled, like a chromosome. So this is when it's kind of like spaghetti strands. It's kind of loosey-goosey. What does that tell you that we're doing with DNA then? We're using it. We're reading it off. We're making protein. We're doing stuff. When it's tightly coiled like a chromosome, we're not using it. We tightly coil it when we need to move the DNA around, the chromosomes around. So in this case right now, we are being used, we're functioning, and then we need to replicate it. Because what would happen if we didn't replicate our DNA and we're trying to make an identical cell? If we had 46 chromosomes and all of a sudden we split it in half, we'd only have 23 in one cell, 23 in another cell. That's when we try to make a baby. That's called meiosis. We're not worried about that yet. This is not for baby making. We need to have 46 chromosomes in the other identical cell. So we have to replicate our DNA so that we can have identical amounts of DNA. We need all 1 through 23 from mom and dad, and all 1 through 23 from mom and dad in both new cells, right? We don't do well when we don't have our full amount of DNA. We really don't do well, okay? And then, so this is when that happens. DNA replication's in the S phase of interphase. And then we get to G2. So this is like the pregnant lady phase. So the cell's getting bigger because we're about to become 2 cells. So this is where we're making like more mitochondria, more ribosomes, more cytoplasm. We need organelles for the new cell to function, right? And we need enough of it for 2 cells now. So we're just growing and enlarging at that point. We're getting ready for mitosis. So I'm going to scroll down a little bit. The pink part is the meiotic phase, and we're going to talk about those details in a second. But first, let's look at in interphase, how do we do our DNA synthesis? How are we going to be able to do it? So DNA synthesis or replication is occurring in what phase? S phase of the interphase section, right? And we have DNA is uncoiling, and we have an enzyme breaking the hydrogen bonds between DNA strands. What does that sound like? We talked about this guy already. We know this enzyme. Helicase. Helicase does this. So helicase does both number 1 and number 2. Remember when we were doing DNA to messenger RNA, we had to open up the DNA in order to read off the message? We used helicase to do that. We're doing the same thing with DNA synthesis, okay? Then we use a different enzyme called DNA polymerase. You know it's an enzyme because it has ACE at the end. And you guys learned when we were making RNA, it was RNA polymerase. So think about the name. You're using an enzyme to make RNA. So what do you think DNA polymerase is doing? You're using an enzyme to put on DNA nucleotides. So T's, A's, C's, and G's, correct? So this is building the new DNA strand. And so the diagram of the DNA synthesis or replication, I wrote it on the right, and notice that I kind of made a fork in it, okay? So I'm going to use a different color to show how we added on. So we had hydrogen bonds between these guys, right? And then helicase came along and he unwound the DNA, and he is preventing those hydrogen bonds from reforming. So we're keeping it pulled apart for a little bit. And if we were to form a new put on new DNA nucleotides, what letter would go on here? C, G, A, T, C. And it would keep going down because helicase would be like sitting here and he's unwinding and unzipping it. And then you guys don't have to worry about the directionality and stuff, but you will in other classes, but not here. So if I have a C on this end, I'm going to have my new nucleotide will be a G, A, T, C, G. And what do I have in between these strands? I have to have hydrogen bonds, okay, just like before. Bless you. Now, the original strand is called the template strand, the template that you're using to create or read off of, okay? And what happens is when we are replicating the DNA, you are replicating it identical unless a mistake occurs because you know what the complementary base pair would be, right? You guys knew what it was. You read it off to me and I wrote it in blue. So can you see that when you pass on this DNA to the new daughter cells, that one side of the DNA strand is the original template and one side is the new daughter strand. So you're always conserving part of it. And then when this gets split up in a future cell, you would split that parent and daughter strand apart and build on top of it. But can you see that it keeps the DNA the same? Unless a mistake occurs, you always have the same strand, right? This strand here is identical to this strand here, right? We have G and C, G and C, C and G, C and G, TA, TA, haha. Anyway, so you guys can see that you have your identical strand, right? So DNA, because it has a T, we know what the other strand is. It's double-stranded still. It's still DNA. We used helicase to uncoil the DNA. It breaks the hydrogen bonds because DNA, between the DNA strands, we use DNA polymerase, okay, because we're putting on DNA nucleotides to make a DNA polymer and bring complementary nucleotides. We now have two identical DNA strands. And this occurs again in the S phase of interphase. So we are doing everything right now to prep, splitting everything apart to have two identical cells, okay? Now, when we get to cell division, so for example, if after we're no longer chromatin and we were to tighten up our DNA into a chromosome, literally what happened was you had a single strand of DNA that I've coiled up in a chromosome shape right here. And if I were to replicate it, all right, you guys familiar with that kind of a shape of DNA? So this is identical to this, okay? Those guys you're going to learn are called sister chromatids, okay? This is a sister chromatid, this is a sister chromatid. Sister chromatids. Okay, so basically, I'm going to come back to that in just a second, but another thing that you noticed, I kind of drew it like with a little waistline. So that waistline is called the centromere. So right here, that centromere, one is holding the two identical strands together right now. Okay, so that's your centromere. And this is also part of what helps us line up the DNA so that when we pull it apart, we know that we have it lined up properly. We know we're taking a 1 and a 1 and separating it so we have the right amount of DNA in the different chromosomes, okay? I mean, different cells, sorry. All right, so cell division. Now we're talking about mitosis. Do you guys notice that these slides here look very familiar to something you probably looked at in lab already? Why, yes, they are. Do you guys remember the whitefish blastula? Yes, the reason we're using it is because it goes through mitosis a lot and frequently. And so that's why we use this as a good example to take a look at it and see the different phases of mitosis. So essential for body growth and tissue repair. There are multiple other additional phases. I don't care about them. I don't want you guys to worry about them. I want you to know the 4 classic obvious phases of prophase, metaphase, anaphase, and telophase. That's it. So there are like a prometaphase and a all these other ones. You don't need to worry about the in-between phases, just the 4 major ones: prophase, metaphase, anaphase, telophase. Okay? So in prophase, what you're looking at is a single cell. They've outlined here where I guess I should put it back in black, huh? They've outlined here the nucleus area inside of the cell. And could you see in other cells that are not going through mitosis, could you see the DNA? No, because they weren't going through a mitotic phase yet. Remember, it's loosey-goosey like spaghetti strands otherwise because we're using it. Now we're starting to condense the chromatin, which is becoming a chromosome. So we're beginning this now in prophase. The nuclear envelope. So remember we had our nuclear envelope around our nucleus, right? It had little pores on it that essentially allowed the RNA to come out, that kind of thing. So it's starting to disintegrate. So what that means is it's getting larger holes in between it. I'll draw it a little better. So it's kind of like it's starting to disintegrate, come apart, disappear, okay? Then you're also going to get what are called mitotic spindles start to form. And this has two centrioles. And you're going to get microtubules extending from the centrioles to the centromere. Do you remember where I said the centromere is? On the chromosome, the waistline part, right? So what we're saying is that it's starting to form mitotic spindles on either end, like on the poles, like a North Pole, South Pole of the cell type of thing. And it's going to have fibers that are going to start to attach to your centromere of your chromosomes. That's what's starting to happen. All of this occurs in prophase. And if you guys look at the nucleus, it's not super obvious. It's kind of, you can kind of still see a nucleus because it's not fully dissolved yet. So those are some hallmarks to know that you are in prophase versus the other ones. If you're in metaphase, this is where the chromosomes line up at the equator of the cell. Another name for the equator is the metaphyseal plate. Starts with an M. Another word for M, use middle. So this is telling you when you see your chromosomes lining up along the middle of the cell, you're in metaphase. So can you guys see how they're lined up along the middle of the cell? So those centromeres are out there. You've got spindle fibers that are coming out to it like this. I'm going to erase this in a second. But it's lining up your chromosomes right along the middle. And for us, that's all 46 chromosomes are getting lined up along the middle, okay? So I'm going to erase that so that you can see the picture better. But another way that you could do this is, so I'm just going to draw like 3 little chromosomes, okay? Picked it. All right, there we go. So if I had 3 little chromosomes with my waistline, they're all lined up along the middle. I've got this microtubules with spindle fibers attaching to my centromere, but I'm all along the middle of my cell. So you could draw like a dotted line for your equator or metaphyseal plate, M for middle, okay? Anaphase is where the sister chromatids are pulled apart toward the opposite poles of the cell. So we said the sister chromatids, that's these guys. Here's one, here's the other, right? So we come back here, and A for anaphase, A for apart, okay? You can also think that when the chromosomes are being yanked apart, they're being pulled at their waistline, so the chromosomes kind of get angled a little bit, and it looks like a sideways A. So everything A for anaphase, right? So for example, it'd be like this. And pretend I'm an amazing artist, right? Okay. They have their spindle fibers here. And also with anaphase, it kind of gets slightly elongated on the cell, okay? Which isn't too big of a deal. But again, if this were the metaphyseal plate or the middle, and you guys can see in the picture here, they're being pulled apart, right? So it's like here, that's what you're seeing, okay? So A for apart, sideways A's if you want to think of it that way, right? Because again, it's like somebody grabbing your belt and yanking you to the side. Okay, and then our last phase of mitosis is telophase. So you could think of this as prophase in reverse. So the chromosomes are going to the opposite poles of the cell where they're being yanked across. The chromosomes are visible but kind of jumbled up, so you can kind of see that. You know, they're kind of there, but they're not so obvious and separated. It starts looking like chromatin again because it can start to relax because we're not going to have to move them around anymore. We already yanked them to the two sides of the cell. And the centrioles will end up going away. Your nuclear envelope is going to start to form again. So it's hard to see on here, but you would start to get a little bit of a nuclear envelope forming around here. The membrane of the cell is like this. And I don't know about you guys, but if you guys noticed, this pinching in here is called a cleavage furrow. Does it not look like what I'm talking about? I'm just saying. All right, so that's another way to remember it. That pinching in part is where in telophase we start something called cytokinesis. Cytoplasm. Kinesis, we're cleaving it apart, okay? You have to get the cell to have identical amounts. Essentially, it doesn't mean you're going to have 5 mitochondria on one side and 5 on the other. Ideally, it would be completely identical, but it does mean you've got to have cytoplasm and organelles, and you definitely have to have the identical DNA replicated, one on each side, okay? So, for example, what do I want to tell you there? So pinching in of the cell into the 2 daughter cells exactly in the middle. So it's usually very obvious to find telophase because you're seeing it pinch in a little bit. And what eventually happens is it will continue to pinch in. It's almost like a drawstring in the middle that's getting tightened up and tightened up and tightened up. And it ends up completely pinching apart and splitting apart like that. And then it's got its own membrane, its nuclear envelope finished forming, and you've got 2 identical cells with identical DNA on either side. You've got proper amount of cytoplasm. So that's the picture I drew here. We had one big cell that we split up, and remember I said the daughter cells are a little smaller because during interphase they're going to grow and become larger cells again. But what would happen if the cell didn't split up exactly in the middle? What if you ended up with one larger one and an itty-bitty one? What would happen? It's divided unevenly, and if you don't have enough organelles or you don't have enough cytoplasm, that little guy is going to die. And that didn't really help you much to replicate your cells if you didn't get another one, right? You didn't go from 1 to 2. You went 1 to 1, and hopefully there weren't other mistakes if that mistake had occurred. What if one cell got all of the mitochondria and the other one didn't get any mitochondria? What happens to the one that doesn't have any? It dies because you need mitochondria to make ATP. If you don't have energy for your cell, it's going to die. Okay, so this has to split up exactly in the middle. It's got to be a good thing. So is telophase part of mitosis? Metaphase, is it part of mitosis? Anaphase? Prophase? Yes, all of those are part of mitosis. Is interphase part of mitosis? No. Is cytokinesis part of mitosis? Not technically. It gets tacked on because it starts in telophase. So it's not technically, it's kind of considered like the afterthought. It's the finishing of the splitting apart. So it's not technically mitosis, but we kind of tack it on there. So, yes?
What protects the DNA while it's being separated?
Awesome question. Why do you guys think he's asking that? What is protecting DNA while this mitosis phase is occurring? Because of nucleases, right? So essentially, the nucleases kind of get turned off or inactivated, and they don't end up working. So they can't chew up your DNA when you no longer have the protective nuclear envelope around it. So it's essentially, you'll learn later on that different ways that we can inactivate our enzymes, our proteins. Sometimes being out of the proper temperature range might cause it. Proper pH range could inactivate it. So for example, like the pH of your stomach, you have enzymes that break down your food. pH of your stomach is very acidic. It's like a pH of 2. And so you can't have a pH of 2 in other parts of your body. That'd be very bad. You'd be disintegrating other things that you don't need to disintegrate. But the enzyme that is active in your stomach, if it ends up getting into other places, is inactivated because the pH isn't proper for it. So depending on different things would allow those nucleases to be turned on and doing what they're supposed to and chewing up RNA. But we don't want to damage our DNA, so it actually gets inactivated. Excellent question. Good job. So, cell aging and death. Dun, dun, dun. All right, so cell aging and death. Harmful agents or mechanical damage. So it makes sense, mechanical damage, like trauma to something could damage a cell. You know, you cut your finger, you've traumatized the cells there. Some of them are going to die because you've just cut them open, right? So that would be like trauma, right? What do you think harmful agents might be? Huh? Shout it out. Acid, heat, UV light, radiation, things that damage your cells. Some of those are obvious to us. Some maybe not so much, but you know, acid, heat, UV light, right? We put on sun protection so that we don't burn our skin. Radiation, you don't want to get x-rays constantly. If you go up in the airplane, you're probably getting more radiation than the typical x-ray you might get once a year, right? But these are things that can damage our cells and stuff, right? And all of those things, both mechanical damage and harmful agents, can lead to cell death, right? And then there's another term called apoptosis. So you would not believe the controversy online of how to pronounce the word apoptosis, apoptosis. I don't care how you say it. Sherry prefers apoptosis. Different dictionaries, medical dictionaries had it both ways. I've heard it both ways from other scientists. Maybe it depends on what part of the country you learned this word. But usually it's apoptosis, apoptosis. Somebody might laugh at you if you say apoptosis, but people say it that way. That just means programmed cell death. So it's the life cycle of the typical cell. So for example, your red blood cells live about 120 days. That's its life cycle. After that, it starts deteriorating. It doesn't live longer than that. It would be really weird if it lived like a year or if it lived 3 years or something like that, right? So there's a natural time limit on some cells. Some live longer than others. And some of them we don't want to die, like heart cells or brain cells, right? So there's programmed cell death where it says this is your natural time limit and you're done. So for example, some of this is occurring like when you are in the womb. So you could have, if you guys look at what an embryo looks like, we have webbed digits like between our fingers and our toes. But when we come out, we don't typically have that webbing anymore. Sometimes we do. So it's natural apoptosis of where that gets disintegrated. Those cells are supposed to die off, and then you come out with your normal looking digits and toes. But if somebody was born where it didn't, what happened? It didn't go through apoptosis like it was supposed to, right? We also kind of have a tail, right? So now you hear like you have a vestigial tail. So you had a tail also when you were this fetus and as you were going through different phases. And then also that kind of went away. Or maybe you have a tail. Some people still have them. And it was because things didn't go the way it was supposed to. Also, if DNA is damaged to where it can't be fixed. So let's say you're out in the sun. You damaged it with UV light. And you damaged your DNA. And now things are going a little haywire. And your cell realizes it can't fix it. That repair enzyme couldn't go back and fix that DNA. It's programmed to die. And so this is also to help prevent cancer. Does everything get caught? No, people get cancer, right? Sometimes this system doesn't always catch everything, or it can't keep up with it, or the damage was too much and it overrides the system, and that stop signal for your cell that should have died off didn't die off, and it's growing and it's growing and replicating. Cancer cells, right? Unlimited growth of the cell that it shouldn't be doing. Okay, so that's the idea of apoptosis. All right, tissues. Woohoo! Let's zip through this as fast as we can. Some of this. So chapter 5, you guys have already seen a lot of this in lab. All right, so tissues are groups of similar cells plus extracellular material that performs a common function, right? So extracellular material is non-cellular parts of tissue, meaning extracellular outside of the cell. So composition can vary. It has, various amounts of water is usually the most prevalent. But we also have protein fibers, which help hold things together. And we also have dissolved molecules, which could be like glucose and oxygen. And this kind of helps the tissues either gel up or have the right matrix, if you will. So it can be fluid like blood. It can be semi-solid like adipose or connective tissue, or muscle is semi-solid. Or it could be solid like bone, right? So it just depends on the tissue type. And you guys we're learning about 4 main categories, right, of tissues, which are epithelium, connective tissue, muscle tissue, and nervous tissue. Those are the 4 tissue types that you need to focus on. And some of the ones that you looked at in lab made sense to you that they were in a particular category, and some of them probably like, what the heck were people thinking to call blood connective tissue, right? So it seemed kind of odd. But as you look at where it came from or information about it, it may have started to make some sense to you. So if we start with epithelial tissue, or we call it epithelium, there are 2 types. Either we're talking about covering and linings, or we're talking about glandular. Could somebody pop that door closed, please? Thank you. So coverings and linings, that means that this is on a free surface throughout the body. Free surface means air is on that free surface side, okay? So that's why on this picture down below, the apical or free surface, there would be air of some sort on that side, right? Gas, whatever is on that side. Versus, we'll talk about in a second. I'm getting ahead of myself. So what would be some examples of coverings and linings? Skin. So you have body surfaces, right? We also have linings of the body or organ cavities. Not a cavity like a tooth cavity, but a space, right? So like an organ cavity would be like the inside of the stomach. There would be a free surface on the inside of the stomach where you have digestion, absorption, and other things going on. You would also have your blood vessels have an apical surface, which would be facing in towards the lumen. Lumen just means like the hole or opening where the blood is flowing through. That would be the apical surface towards that opening. Apical surface of your skin, in your mouth, it's kind of in your body, but it's an outside type surface, but it's lining the mouth, lining the trachea, lining the esophagus, things like that, okay? Let's see, glandular forms glands of the body. So glandular cells are epithelial cells in appearance and are what they originate from, thus that's why glandular is under epithelial. So special characteristics, you have cellularity, which consists of tightly packed cells. Well, that makes sense when we're talking about skin, right? We don't have big bubble holes in our skin. Typically, I guess you could have blisters. That's not good. I don't know. Specialized contact. So cells fit together to form sheets. That makes sense for protection and stuff, right? You want sheets of cells tightly packed together to form this, what we think of as our solid surface of skin. There is polarity, meaning it possesses 2 different surfaces. So we already talked about the apical surface, meaning the superficial or free exposed surface towards the air side. The other side that I didn't mention yet was the basal surface or basement membrane would be on the basal surface. So here's the basal surface, which is this end here, and this would be the basement membrane. So this would be the apical surface, this would be the basilar surface, okay? And on the other side of the basilar surface is typically connective tissue. So something is on the other side of the basal side. It's not the free side. Okay, so deeper attached surface. And let's see. Attached to a basement membrane, that would be for the basal surface, and associated underlying connective tissue, which we said is right underneath it typically. And then we have something called avascular but richly innervated. And so what do you guys think avascular means? A usually means not. So vascular, not vascular. So lack of blood vessels here, no blood vessels. Okay, versus, but it says it's richly innervated. So what do you think innervated means? Lots of nerve fibers going through here, nerve sensation, right? So no blood supply, but lots of nerves. So you can feel pain here, right? Yeah, okay. So let's put, no, I abbreviate blood BLD. I know I'm just skipping 2 O's, but no blood supply, but good nerve supply. Okay, so for regeneration, high rate of cell division. So for epithelial, this makes sense, right? Because it gets bumped up, abraded, scraped constantly. Even in your mouth, it does, right? You put food in your mouth, you chew, you bite yourself sometimes. If you chew chips, you know, it scrapes your mouth sometimes. You kind of feel a little raw after you've eaten some foods versus others, right? So it makes sense that we need to regenerate that tissue frequently. So epithelial cells typically have high mitotic rate, okay? So it depends on where you are, but like the lining of the gut might turn over about every 3 days, and your skin, though, could take maybe a couple of months to totally regenerate and stuff. So lots of mitosis. Lots of replication going on there. And classification of epithelia. So you guys kind of talked about this in lab already. They will classify it by shape of the cell and the number of cell layers, okay? So basically, you have 3 basic forms. We have squamous, cuboidal, or columnar. Squamous means thin and flat. So the word squamous is Latin for scale-like. So we're talking about like scales, like lizard scale or skin snake scales. That's thin and flat, right? So it's scale-like. And if you were to take a cell and cut it in half and look at the side of it, they say it kind of looks like fried eggs. So it's kind of like got this little bit of a shape to it, a nucleus in the middle. So it kind of looks like a fried egg from the side. Let's see. There's not usually a lot of cytoplasm in squamous cells, okay? And if we look at a cuboidal cell, it says it's kind of shaped like a tiny sugar cube. I kind of think they almost look like little square balloons because they're kind of poofy a lot of times. And it's never taller than it is wide. So never taller than its width or wide. And if I were to draw a cuboidal cell, it'd be kind of like this. And it usually has more cytoplasm in it versus a squamous cell. And if we talk about, it doesn't look like fried eggs either, lots of cytoplasm. If we talk about a columnar cell, I always think column-like, like a tall column. So it's a rectangle, tall shape. So shaped like tiny columns. So these are taller than wide. Okay, I don't think I have to draw a rectangle for you. I think you guys get that. So classifications by number of layers. So if you say that something is simple, you're talking single layer. If you're saying stratified, you're meaning multi-layered. It means 2 or more layers. And I know sometimes when you guys were looking at some of the histology slides, it wasn't always easy to tell if something was 2 layers like on top of each other or 2 layers next to each other. So it's not always obvious, but if you are told what type of tissue it is and then you kind of understand, oh yeah, that's typically stratified cuboidal, blah, 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 or something like that. Stratified squamous is usually pretty obvious, especially if you see a free apical surface, like you're looking at skin or something like that. So simple epithelia is single layer, and it is best for filtration, absorption, secretion. So it's telling you these are thin, flat cells that filtration, secretion, absorption, things are passing through them, right? So diffusion, osmosis also would fit under filtration, okay? So if we talk about simple squamous is really good for, I already said that. All right, so some examples where you will find simple squamous would be like capillaries, okay? Small air sacs of the lung, where you have gas exchange occurring, right? Lining of glomerulus, which was in the kidney. And you guys were supposed to look at the kidney glomerulus. It didn't matter what your textbook was asking you to look at, but we wanted you to look at the kidney glomerulus. And that's what this picture is here, this histology slide. This area, all of this roundy stuff within that space is called the glomerulus, which is where filtration, where your filtrate or your urine is first starting to be filtered and produced. That by the end, it becomes your filtrate or your urine that will exit. But it is coming through the blood vessels, and things are passing through that lining into the glomerulus, and then they're going to come out into this space and end up coming down the various distal and convoluted tubules. You'll learn more about that in 228. But so what you wanted to see, the squamous cells that they were showing you were lining the glomerulus. So these are all squamous cells. Can you see how they kind of look thin, flat, not a lot of cytoplasm? They're lining that membrane around the glomerulus, okay? And let's see, what else do I want to tell you? Also, lining of blood vessels. Lining of BV is blood vessels. And the lining of blood vessels actually get a special name, and it's called endothelium. It's still epithelium, but it's a special name because it's blood vessels, so we call it endothelium, okay? So that could be a capillary, but it also could be the lining of an artery, the lining of a vein, so on and so forth. We will pick up, I think, so we have enough time for the clicker. We'll pick up with the rest of simple cuboidal and columnar and then get into the pseudostratified and goblet cells and all that stuff on the next lecture. So